All right, good morning, everyone. My name is Aijun. So I know most of you. I'm a Ph.D. I'm working in the lab, surgical bioengineering lab in research too. So I came to UC Davis in 2012, so it's a little bit over 4 years already. So today I'm going to talk to you about the lab. So in the past, a little bit over 4 years, we have grown from a very empty space to a pretty well-established lab with quite a few useful tools that I think, for your practice, for your research interest, if you would like. So I'm gonna talk to you about the research projects and also the tools and technologies that we have available in the lab that potentially we can help you with for your research interest. So the surgical bioengineering lab, as I said, is in research 2, the 3rd floor. We have quite a few faculty members there, Dr. Farmer, myself, and Dr. Perez, Dr. David Sahar, and Dr. Ian Brown, or in the research 2 building for research. So for my own research interest, I was trained in bioengineering at UC Berkeley doing my postdoc. So my research interest is mainly focused on tissue regeneration by using bioengineering tools and technologies, especially stem cell engineering to make some biological stem cell product or therapeutic, and biodegradable polymer or extracellular matrix scaffold-based biomaterial engineering. So the ultimate goal is to combine biomaterials and stem cells for different disease treatments. So currently in the lab, we have basically 3 different arms for doing the different research projects. The first arm is biomaterial. So we're doing a lot of scaffolding, surface modification of devices, 3D structural fabrication for a scaffold, and I'm going to talk to you about that later. And stem cells actually is the main research focus in the whole lab, especially stem cells from different sources. So especially stem cells from induced pluripotent stem cells and stem cells from perinatal tissues, different tissues such as placenta or other perinatal tissues. And we have a large arm of animal modeling for different human diseases. So I'm going to talk to you about that too. So we are focused on regenerative medicine, which meaning we want to regenerate tissue or tissue damage. We want to interrupt or want to improve the standard of care. We're doing translational research. We're trying to build up some products down the line so that we can use for clinic. So hopefully today you will find something that's interesting to your practice or research interest, so that you can always come to the lab and talk to me or other lab members and see what we can do for you. So a few examples what we can do for surgical conditions, surgical diseases. Number one, that's our major research focus right now in the lab, is some bioengineering approaches for birth defects or fetal surgery. So as you may know, Dr. Farmer and I are co-PIs on a large CERN grant supporting our spinal bifida research. I believe most of you have seen the video, but I'm going to show you that again today. So just to show you what we can do by combining surgery with bioengineering approaches and how we can do things differently. So for example, other applications like peripheral nerve regeneration or peripheral nerve injury, and you want to, especially for large defect, large gap that you cannot directly suture them together, that how you can use some devices to help the tissue regeneration. And the third one was just very commonly seen spinal cord injury, for example. So when the spinal cord tissue is damaged, it's usually very difficult to regenerate. So what we can do in the lab is to add some biological components or some biological tools to help the body to regenerate better the cord tissue, or to protect neurons from being damaged, further damaged, something like that. I'm going to talk to you about the stem cell related products in a little bit. And also cardiovascular diseases, for example, or so for blood vessel regeneration or bypass blood vessel, can we use bioengineering approaches to make a better bypass product that tissue can, like especially endothelial tissue, endothelialization can happen faster or better to cover the surface that blood is contacting, so that once the endothelial cells is covered on the surface, like sooner than the thrombosis or other bad effects happening, the patency will be improved. So these are just a few examples, but we can do a lot of other things too. I'm just trying to stop here for a little bit and think about what's your research interest, what the bioengineering tools and technology. I mean, I can only cover very little bit today, but there are so many things happening in the engineering world and bioengineering especially. So like a cell expansion, culture, in vitro, all those things, and devices, fabrication, 3D printing, surface modification, just to make your device more personalized or more your disease specific or your application specific, more effective. And also some devices for example, bioreactors and device, fabricating capability, and also a lot of animal modeling for your specific research interests. I think we can probably help. So let me start from stem cell part. So as I mentioned earlier, so we have many different sources of stem cells. Today I'm going to talk to you about two main cell sources that we can use for research and for future applications. Number one is as you all know, the autologous cells might be the best cells for the patient specific applications. So how can you get autologous cells for different type of cells? If you think about this for a minute, it's not easy, right? For example, if you want to get some endothelial cells from a patient, it's not easy to get endothelial cells. You have to cut probably a segment of a blood vessel or somewhere that you can sacrifice, get tissue, and cell isolation. Those kind of process, very difficult. But there's a new technology called reprogramming, so which is I believe most of you would know, IPS technology, induced pluripotent stem cells, which is to induce patient fibroblasts, for example, or any tissue biopsy cells that you can use lentiviral system or other transduction system to make these fully differentiated cells into a embryonic-like cell stage, and then you can differentiate those cells into the cell type that you want. So any cell type, basically, all the cell types that you can see in the body. So today I'm going to talk to you a little bit about some research that we're doing on here, which shows the promise for making patient-specific cells, different cell types, for any specific disease treatment. For example, endothelial progenitor cells that we can differentiate from human iPS cells. So human iPS cells can be generated from any skin biopsy, urinary sample, many fully differentiated cells. That's very well established. It's a 2012 Nobel Prize science, actually. It's very well established now in the lab. And neural stem cells that we have been working on for quite a few years. So it's another type of multipotent stem cells that can differentiate into multiple lineages. So can use that for different tissue regeneration purposes. So another portion is perinatal tissue, especially for our birth defects or congenital diseases. So from perinatal tissues, placentas especially, I'm going to talk to you about that, how you can generate stem cells from placenta at different gestational stages. And also there are some other tissues available for cell derivation, for example, amniotic fluid samples. I'm going to talk to you about that too. So another one is very well established in the lab as well, is umbilical cord blood sample. So if you get some umbilical cord, you can get the blood sample, you can generate a lot of very young and very immature progenitor cells, stem cells, for example, endothelial progenitor cells for your different applications. So based on all the stem cell or cell research, we are trying to establish some stem cell or cell product. So either a cell product that you can use off the shelf, or you can process the cells with some ECM or scaffold. So for different applications, you can ship to some other places. That's a hope for cell product development. On the other side, we can develop some cell-derived products. So it's not live cell product, it's a cell-derived products. For example, cell secreted stuff in the culture media that we can isolate or concentrate. So conditioned media, for example, or exosomes. So that I'm gonna talk to you about that in a little bit as well. It's like growth factors or exosomes that are produced by cell culture. So you can culture a lot of cells in vitro, expand many flasks, and then you collect the cell conditioned media, and then you extract or purify the components that you want to have for specific applications. So first, I'm going to talk to you very briefly about the differentiation of human-derived iPS cells into the cell type that you want to use. So as I mentioned, this is the process for reprogramming. It's very simple, very straightforward. You have some adult somatic cells you can easily get from patient biopsy, skin biopsy, for example, and then culture these cells from the skin biopsy sample. You can reprogram these cells. There are so many different ways nowadays to reprogram cells into iPS cells, like a embryonic stem cell-like stage, so that are pluripotent, meaning they can differentiate into all the cell types, all the lineages in the body. One direction that we can go is to use pluripotent stem cells to differentiate them into a neural crest stem cells. So why would not be optimal to use iPS cells directly for therapy? If you think about this for a little bit, because these cells are pluripotent, they are very, they can form teratoma very easily. They're growing very fast, and if you transplant these cells directly into an animal model, for example, so they will develop teratoma very fast, and they are fast proliferating. They're also making all different type of tissues that you cannot basically control them very well. But once you differentiate these cells into certain stem cell, instead of pluripotent, maybe multipotent or unipotent progenitor cell stem cell lineage, that means they can only go to certain pathways instead of going into many other different pathways. So that way you can control the cell fate basically to a range of the differentiation potential. So this is one example that neural crest is a very important transient developmental stage. So it's very difficult to get neural crest cells from the embryo. You have to destroy embryo at certain developmental stage to get those cells. And they are multipotent. They can make ectoderm tissues, and they can also make mesoderm tissues. So they're multipotent. They're very important for making a lot of the tissue in the adult stage. But it's very difficult to get them from development. So, but using iPS technology, you can establish a differentiation protocol that can control the iPS cells to go into neural crest stem cell stage very specifically. For example, here we've identified a protocol that you go from iPS cells through different stages, and then you get monolayer. So iPS cells, like embryonic stem cells, they are cultured in clones, so they can see very well-defined colonies. But once they differentiate into certain multipotent stage, they will, like for neural crest stem cells, they're monolayer culture. They're not making any teratoma in the in vivo applications. And we also show these cells differentiate from iPS cells. They're multipotent, just like embryonic neural crest stem cells. They can make neurons, they can make Schwann cells, they can make all these mesodermal lineages, chondrocytes, osteoblasts, adipocytes, smooth muscle cells. So if you stop here for a little bit, you can think about, you get the skin biopsy from a patient. Actually, you go through reprogramming and differentiation. Now you get all different type of differentiated cell that you want to use for this patient. It's patient specific. There's no rejection. There's no immune rejection that associated with allogeneic, like from another donor, for example. So there are many applications that we can use for the reprogrammed iPS cells and differentiated cell types in combination with a lot of scaffold technology that I'm going to talk to you about in a little bit. So you can do stem cell transplantation for different disease treatment. So this is one of the diseases that we worked on, which is a peripheral nerve regeneration. So it's in the rat sciatic nerve injury model, we made a 1 centimeter gap, which is a very long gap for a little rat. So without anything, that if you don't do anything, just leave the gap there, the rat cannot regenerate the nerve tissue. But with the bioengineered scaffold and stem cells, actually we can see here, so you can see both in both groups. You see some in the middle of the nerve conduit. You see some tissue regeneration in the middle of the conduit. But with human stem cells, neurocrest stem cells, which is a Schwann cell precursor cell, actually. So in the immune deficient rat model, you can see many more myelinated axons, which are contributed by these transplanted cells, as you can see here. So the green is S500 beta, which is a Schwann cell marker, and human antigen is shown here in nucleus is a human pneuma. So which means, in the cross section, or this is actually a longitudinal section of the nerve conduit. In the middle of the section, you can see actually the aligned axon regeneration, especially all these green tunnels, which are myelinated axons. And human cells actually participate in this regeneration process and making all those myelination structure. So if you do some electrophysiology at 1 month after surgery, you will see actually with stem cells plus all the delivery vehicle, the conduit and matrigel or hydrogel delivery vehicle, you will see a very significant electrophysiology outcome. Versus in the conduit, empty conduit without stem cells, you don't see that. So this study means we can generate a multipotent stem cells. In this particular case, is a Schwann cell precursor cell. You can transplant these cells in a nerve regeneration model, and these transplanted cells can help peripheral nerve regeneration by producing more myelination. So which helps actually the axon elongation and helps the electrophysiology outcome. So in this rat model or immune deficient animal model, rat or mouse models, you can look at human stem cell function. So all the immune system is blocked, and the human cells will behave in the way that they would behave ideally in the human transplantation cases. So this is another example, very briefly. We can also differentiate iPS cells or embryonic stem cells into endothelial progenitor cells. This is a collaborative project with Dr. Jan Nota's group, Dr. Ping Zhou, an assistant professor in the stem cell program. So we're deriving human-specific iPS cells-derived endothelial progenitor cells. So you go through, very similar to the other study, we establish a protocol going through the differentiation process. You get the endothelial progenitor cell at the last. So all those cells, they show very pure endothelial markers like CD31, VWF, and also they can make tubes, like tube formation, and they can uptake LDL. And in the animal model, transplantation model, we actually found that human CD31 is a human stem cell marker, a human endothelial cell marker. They form some blood vessel structure this was done in a NSG immune deficient mouse model, subcutaneous injection of a hydrogel loaded with iPS cell EPCs. So these cells are able to make endothelial cell structure in vivo and make blood vessels. So this is not a disease model, but it shows the potential of those iPS cell derived cells. So if you think about from the top to the end, you actually get some human-specific iPS cell-derived endothelial cells. You can get iPS cells from many different sources, as I mentioned earlier, skin biopsy, for example. You can get EPCs from skin biopsy, basically. And you can use EPCs for many different applications. So the second portion of a cell source is a perinatal tissue that we have been working on for the past over 5 years. So placenta especially is usually discarded when the baby is given birth. But placenta tissue is also available from different gestation ages by biopsy, actually. And amniotic fluid is another source. So if you think about how the baby is located in the mom's womb, so there are two sources of fetal cells that you can get without disturbing the fetus. So one is a placenta biopsy, the other one is amniotic fluid amniocentesis. So both these samples can be obtained at birth, but you can also get those tissue samples from early gestation. So for placenta especially, we're very interested in looking at the stem cell looking at placentas as a source for stem cells. So as I said, discarded term placenta or donated placenta mid-gestation, early mid-gestation, or chorionic villus sampling with CVS is a first trimester placenta biopsy technique that is usually used for fetal diagnosis. So we can actually, we know that placenta is very important for development, and placenta during the whole gestation stage, they are very important in making many different things for development. So for example, there's a paper published in 2012 about this serotonin secretion from placenta. So if you block placenta serotonin source, actually the fetal forebrain development will be blocked. So in our lab, we have established a protocol to isolate mesenchymal or stroma cells, mesenchymal stroma cells from different gestational stage placentas. For example, this is a CVS age placenta, which is about 8 to 11 week gestation. Although the CVS tissue is very, very tiny, it's only like 30 to 100 milligram tissue, but we have established a protocol to expand cells into many millions of cells within like 3 weeks. So these cells are unique. They have many unique properties. Number one is that these cells are fetal origin, so which means you can use these cells autologous as an autologous cell source for fetal treatment or postnatal treatment. So it's a very unique autologous cell source. And they're fetal, meaning they are very early in gestation, in development as well. They are very expandable, and they are secreting a lot of very important factors, growth factors, cytokines, very important for development and for healing. As we all know, fetal environment is very pro-healing, and all those placenta-derived early gestational stem cells, they are actually very important. They're very secreting many very important factors. For example, we're looking at BDNF, brain-derived neurotrophic factor, is a very powerful neurotrophin, very important for brain development and spinal cord development in development, and also neural survival. So one thing that we discovered, that many in vitro work has studies have been done in the lab. One thing that I want to mention today is the BDNF secretion level. Actually, if you look at adult bone marrow MSCs, they're all MSCs. So you can generate MSCs from different sources, like adult bone marrow, that's very, very often done. And our stem cell program here has a fantastic bone marrow MSC product, a series of products for different diseases. But if you compare placenta MSCs with the adult bone marrow MSCs, you see much higher level of BDNF secretion in the placenta MSCs than adult MSCs. So one thing that Dr. Nota, our stem cell program director here at UC Davis, one project they're doing is to make adult bone marrow MSCs to secrete high-level BDNF for Huntington disease. What they're doing is to transduce bone marrow MSCs with lentiviral vector to overexpress BDNF. So actually, in our case, the placenta MSCs are naturally secreting high-level BDNF. So you don't need to do any gene delivery or virus manipulation for those cells. Potentially, these cells are very useful for many BDNF-related applications, brain, spinal cord, many other neural survival-related applications. But this is just one side of it. Many other factors are secreted by placenta MSCs, actually, that you can, like VEGF is pretty high level as well. And HGF is also very highly expressed in placenta MSCs. So we have transplanted these placenta MSCs in the fetal sheep model of spina bifida. That this is a major collaborative project between Dr. Farmer and myself and the whole lab, surgical bioengineering lab, supported by CERM and the Schreiner grant. So basically, we're looking at how we can supplement the fetal surgery. As you all know, fetal surgery can actually treat spina bifida very well before the baby is born. But can we do that better? Can we supplement the fetal surgery with stem cell products and make these patient outcome better? So we have this. We just transplant these cells into this animal model at the secondary surgery, which is that this model is the first surgery at GA75, and then the second surgery is the GA100. And then 45 days later, the spina bifida baby lamb will be born. So when we transplant cells here at the second surgery, when we do fetal surgery, we add a component which is stem cell product. So in this particular case, we have placenta MSCs co-cultured with a ECM matrix, a scaffold sheet that we can patch the defect with. And so here, actually, I'm going to show you a very short video that I believe most of you have seen that before. Is highlight the importance of the stem cell function in this fetal regeneration environment. When you have, so we have two twins. One was treated with stem cells plus matrix. The other one was treated with a matrix only, without stem cells. As you can see, right at birth, this is the beauty of the lamb model. Right at birth, the lamb would be able to stand up and walk if they are able to. So in the spina bifida lamb, they develop very typical paralysis associated with spina bifida. This is the gold standard animal model for spina bifida research. But this lamb was treated with the stem cells plus matrix. So the other one was treated with matrix only. So we have seen, this lamb actually was able to stand up and walk just normally. And the other one, they was developing very typical paralysis associated with the spina bifida. And this stem cell treated animal actually was able to stand up and walk in coordination, very nice coordination in all the limbs and tail movement and nurse, everything is just normal. So we've done this many sheep actually now. I have more numbers than this now. But generally speaking, we found that this recovery or this stem cell mediated regeneration effect actually is a very reproducible and consistent. So we hope that these cells are able to, we're able to develop these cells as a product in the future. So we did some histology. If you look at, this is the normal spinal cord. If you look at the butterfly structure with the gray matter, where all the neurons are located, you can see a lot of very normal phenotype of large neurons there. And this is the skin repair, which is a mom's trial, skin repair, fetal skin repair. You see some preservation of the cord tissue, but very, very few neurons that can survive in the whole process, actually, at least in this animal. And so this one is the ECM scaffold only without stem cells. You see some neurons, actually, which means the ECM scaffold is plus fetal surgery actually is helping the recovery in the spina bifida. But not as good as this group. This is the ECM plus stem cells. In the gray matter, you'll see large density of very high density of neurons actually survive. And vehicle plus PMSCs, you see the large neuron density is much higher than the vehicle only. And you see a very pretty linear correlation between the neuron density and how well the animal was able to walk, which means the better neural preservation, the better the animal will be able to walk. It's very, very important. It shows the importance of the stem cell function in this particular case, neural protection or neural preservation capability. So now we're still doing a lot of sheep now. We are basically trying to figure out the dosing. We're trying to figure out how to deliver the cells better, what delivery protocol or product is the better. And also we're developing protocol to make a cell product. So we're using all these GMP, good manufacturing practice associated procedures, reagents. We're making a cell bank, basically, that can potentially be used for off the shelf for different patients. So that actually is going on very actively in the lab right now. We're developing many cell lines and many vials. So hopefully at the end of our current CERN project, it's a preclinical development award. So at the end of this project or close to the end, we're going to apply for another grant. Hopefully we can get a translational clinical research grant so that the stem cell product can be applied into some clinical applications. So as I said earlier, we're also making a lot of cell products, like cell ECM, co-cultured cell ECM as a patch material that can potentially be used for many defect treatment. We're also looking at the stem cell secretome, stem cell secretion, growth factors, or other things like exosomes. So we're looking at the ECM loading and secretion, so how the extracellular matrix scaffold, the clinical grade ECM product, how the stem cell can be loaded on the ECM, how they behave in vitro and in vivo. So one very important thing that I think, to my opinion, is some in vitro model to test how your cells are. So instead of using a very expensive animal model like the sheep model, if you have a platform that you can use in the lab to test which cell line is better, so that would be fantastic. I mean, everybody is trying to establish some in vitro cell screening model. So in our lab, we established a neuroprotection model. Neuroprotection is considered one of the major mechanisms for the spina bifida fetal surgery repair. So as we can see in this model, basically we develop a neuron blastoma cell line. Actually, it's a human neuron cell line. You culture these neurons here, and then you damage these cells. And then you apply different cell lines and see which cell line can protect the damaged neurons better. So for example, we see very consistent protection between cell lines without PMSCs. Generally speaking, with PMSCs, placenta MSCs, more neurons will survive. They've developed better neural network. But between different cell lines from different donors, different cell lines from different gestational ages, the protection effect is different. So we're using this as a in vitro model to screen cell lines, basically. So another thing that we're doing very actively in the lab is the stem cell-derived exosomes. As you may know, so exosomes are some nanoparticles secreted by cells, and that can mediate cell-cell interaction. That's how the cells that are apart, very far away from each other, how they sense the other cell communication or the other cell function or need or those kind of things. It's like a cell phone or a smart cell phone. That's what Dr. Jan Nota's group has been calling exosomes. Like you call the other cell far away from you without giving direct touch, but you can give these information by exosomes. So exosomes are secreted normally by cells, and these exosomes can be taken up by other cells. So different cells in different environment, different cells may pick up different exosomes. But in our particular case, we want to develop stem cell exosomes. The main reason is based on our sheep study that I didn't mention earlier. So from histology, we noticed that there are no human cell engraftment at 45 days after transplantation, which means, but the lambs were able to walk. So what's going on between these 45 days? We have done multiple time points lamb histology, but basically we concluded that the transplanted human placenta MSCs, they don't stick around long. They don't stay there for long. My guess, I don't have a very firm conclusion on how many days exactly they stay in the environment. My guess is about 2 weeks, 2 to 4 weeks. But they disappear over time. But they've left a lot of very important factors or stuff where they stayed. Exosome that we believe is one of the major product that the cells secreted, and then they're gone, they leave. But they left those important components there. All those exosomes secreted by the transplanted cell, they can be eaten by other surrounding cells, for example, neurons. So we have shown this actually in vitro in the lab. We have a large-scale culture, PMSC, placenta MSC culture. We established a protocol to isolate exosomes from culture. They are nanoparticles. You can use many different ways to isolate these stem cell exosomes. You see very, very many proteins actually are contained in exosomes. You see some other very important proteins, for example, galectin-1. So I can't go into any details for here, but there are many important studies about galectin-1 in stem cell transplantation applications. So these exosomes, stem cell exome, they are actually expressing high-level galectin-1. And there are many other integrin, very important cell surface protein are expressed on the exosome surface, which means that's how the exosome, stem cell-derived exosome, particularly here, how the exosomes are taken or eaten by other cells by cell-cell binding mechanism, basically. So we have one project very actively going in the lab so you can generate a lot of stem cell exome. You can directly use these stem cell exomes for many diseases because they are nanoparticles. You can inject IV, you can locally put those stem cell exosomes anywhere that you want, because there's no live cells related to this application, to this end. So there are many other research projects going on about stem cell exosomes for different disease treatment that we just don't have the manpower in the lab to do that now. But what we want to do is to engineer these stem cell exosomes and trying to make these stem cell exosomes more specific. For example, based on the surface marker expression, integrin expression, you can engineer and add some ligand to the exosome surface, make them different so that they can be eaten by certain cell types more specifically. If the ligand is specifically binding to endothelial cells or neurons, so these stem cell exosomes will be eaten by neurons or stem cells or apoptotic cells or other cells just based on your need or your design. So they can be more directly eaten by certain cell type, so that the effect will be hopefully higher, the better, because the precious stem cell exosome. So stem cells are making exosomes but pretty low level compared with the cancer cells. But how you can use these stem cell exosomes more effectively, that's a key thing. So we're developing this. Basically, you can use these things for different applications. You can rescue neurons, you can rescue endothelial cells, you can target immune cells. You can do many different things just based on your design. Very briefly about ECM. So extracellular matrix, as you know, the body is made of cells and extracellular matrix. So we're talking a lot about cells, but at the same time, and on the same side, the other side, there is a cell extracellular matrix, so where the cells are living in. So you can get a lot of native ECM. Native ECM can by decellularization. You can also get some artificial ECM that you can produce in the lab, or in a production site, to make artificial ECM that mimics native ECM as much as you would like. And also the functional ligands that can be used to modify artificial ECM. So artificial ECM, you can make them into different shapes, but they don't have any biological stuff. Then you can engineer the surface to make them more biologically active. So ECM, as I mentioned, earlier in the sheep study, actually we're using a Cook product that Dr. Boyd actually is an expert in, the decellularized SIS product from the pig. And so they're making the decellularized SIS into different formats, the sheet of material or many other formats like hydrogel or emulsified injectable format or some other things. And in our lab, actually, we are developing some human ECM. So as you know, all those ECM mainly are produced by pig tissue, but they're from different species, right, from pigs. There are some rejection problem associated, may not be very significant, but ideally human ECM may be better. The reason we're looking at this is because we have lots of placentas in the lab, and placentas are human or like a tissue, and they're discarded usually. So we developed a protocol to isolate chorionic villi and isolate amniotic membranes, and we decellularize this tissue into ECM. So we have developed a protocol, as you can see, very well decellularized, after decellularization, before decellularization. And we can also develop this into an injectable hydrogel that actually one of our research residents, Ben Keller, has been very actively involved in this project, and another graduate student. So we can also use these injectable hydrogel, human ECM hydrogel, for human cell culture expansion or delivery. So potentially we can use this as a product for many stem cell culture or expansion or delivery. And we also noticed that these human ECM hydrogel, they are bioactive. They're not inert. They have lots of good stuff still in there. So the protocol is pretty mild. Most of the growth factors that we can still detect afterwards. A lot of angiogenic cytokines are still present. And we did some in vivo initial testing that we're going to be doing more to confirm this. But in the initial study, we found that actually, compared with the bovine tendon collagen, as you can see, like 2 weeks after in vivo transplantation, you see the tendon-derived collagen, bovine collagen, you see many more cell infiltration, many more cells infiltrating into this collagen hydrogel. This is a hydrogel, this is a hydrogel. At 2-week time point after transplantation, you see many more cells inside that collagen hydrogel. So it's a body response. I mean, it's very normal. Whatever you put in the body will react to the implanted scaffold or material device. But in the amnion hydrogel, that actually you don't see that many cell infiltration. A lot of cells are on the surface, but they cannot go very deep. And we also want to do a translational research on this product. We use an anti-adhesion model in the rat model. So we have 2 different products. One is a sheet that right after decellularization, you have a sheet membrane. And the other one is an injectable hydrogel. So this is the model that we created in the lab, and it's pretty well established. So without any treatment, the abdominal adhesion will form on all these buttons very significantly. But when you have a coverage of the amnion membrane, actually the adhesion is inhibited in the buttons that covered by this amnion membrane, not on the other ones that not covered by the amnion membrane. And injectable material is even easier for application because you can just smear it all over the place, and they actually inhibit the adhesion formation quite significantly. We don't have very many cell number at this animal number at this point, so we're gonna repeat this experiment, and we're gonna do more research on this. I think it's a very important product. So I think I've shown this. I don't know. I have like a few more minutes, but very briefly about the artificial ECM. So I want to show you what we can do in the lab. You can make some native ECM, but you can also make some product on your own. So one very powerful tool is electrospinning. We have a machine in the lab that you can use to spin polymer into nanostructure, nanofibers, for example. You can make these nanofibers align in a certain direction. And the cells actually can sense the alignment. They will behave differently on the aligned nanofibers versus random nanofibers. So we can make all these nanofibers into different shapes, like conduits. You can make a line on the longitudinal line in the luminal surface, for example, and circumferentially a line on the outside of the scaffold, for example, for blood vessel regeneration, just to mimic the tissue that you want to replace or the disease application you want to treat. So we've done something for the spina bifida application, actually, for spinal cord. This is done by Pam, actually, quite a few years ago when I was still Berkeley and he was at UCSF. We can use the same technology to make different shapes. So for example, for the spina bifida or spinal cord regeneration scaffold, you want to make the longitudinal aligned fibers in the luminal surface so that we're hoping the axons will grow longitudinally so that they can connect with other neurons more efficiently. On the outside, you want to make this kind of directional fibers so that the suturing capability is higher. The mechanical strength is higher. But you can make many different applications. This is a very recent data that we obtained. By using these aligned nanofibers, we have myogenic cells, muscle cells. When you culture muscle cells on the aligned nanofibers, the alignment can actually help cell fusion to make a more powerful cell myogenic population or muscle-like structure. So, and I'm going to skip this. This is very advanced, actually. I wish maybe next time I will talk to you more specific on this project. This is a high-throughput screening system that we can use to identify specific ligands for specific cell type based on the cell surface marker, especially integrins. In this particular case, we're using endothelial cells as a model because endothelial cells are very important for tissue regeneration, angiogenesis, wound healing, everything. So we know surface marker expression on endothelial cell or many other cell types. Then you can use one compound. Is the high-throughput screening technology developed by Dr. Kit Lam on this campus, actually, in the biochemistry department. So we have been collaborating on this project for a few years now. We developed quite a few very powerful ligands that we can use to modify scaffold. So I won't go into here, but based on some chemistry, you can present a ligand on the surface of a inert material so that the inert material will become more biologically active. And you can design the surface, make them in the way that you want them to be. For example, endothelial cells, with this ligand, actually, you can see much higher cell binding affinity on the modified surface compared with the inert material or the unmodified surface. This is a in vivo animal model in the rat cardiac artery bypass model. So you can see the ligand-modified artificial vascular graft, they are more patent than the non-treated control vascular graft at a certain time point. So last slide, basically, about animal modeling. So I will talk to you more about the ligand. I think that can be very useful for many different projects. The ligands can be used for different cell types, but I would have to skip that. So these are the animal models that we're using in the lab. So when you have a product, before you move into clinical trial, you have to do some in vitro functional assay and in vivo in animal models functional assay. We have mouse in utero transplantation model. So in the mouse model, you can transplant stem cells or anything that in utero before the baby mice are born. And we have the NOD-SCID gamma, which is an immune-deficient mouse model, very widely used for human stem cell transplantation. And to look at the human cell fate after transplantation, we have a rat spina bifida model, retinoic acid-induced rat spina bifida model. We also have a rat CDH model. We have a rat abdominal adhesion model that I mentioned earlier. We are going to establish a guinea pig model for fetal research for many reasons, and we're also developing some very interested in collaborating with the vet school on developing some naturally occurring disease model instead of just the surgically induced animal model. And of course, we're still doing a lot of the sheep model. We have the pregnant sheep and spina bifida lambs, and we're doing research on that. If you have any interest in any of these for your stuff, just go ahead, let me know. I think we can help. So in conclusion, I hope I have confused you enough today. There's many different things going on I was trying to cover. But I think I want to tell you, what I understand surgical bioengineering and what we're doing in the surgical bioengineering lab here at UC Davis School of Medicine. So what bioengineering can be used for all this surgical disease treatment is what we call surgical bioengineering. All the available bioengineering tools and technologies that can be used to improve surgical practice, to make your product better or make a new product. So, and also we're hoping, and as you see, for example, the sheep spina bifida model for fetal research, fetal treatment. The bioengineering approaches in that particular case is a ECM cell co-culture, so the cell secretion can be monitored or can be changed, or maybe can be changed. So we can use these bioengineering approaches to improve the standard of care for many disease treatments. And also all these, we have so many tools available in the lab and technologies. If you have any needs or interest in any particular thing, just go ahead, let me know. I think we're interested in doing more surgical innovations for any of your project or interest. I think now we are mature enough, we can provide a lot of consultation or technical support. So what we do actually in the lab is trying to just bring MDs with the Ph.D.s or technical bioengineers just together so that we can do our practice better in the future or now. So all we do is the translational medicine. Any need, I like to talk to many of colleagues about ideas or need that you want to do. So I think our lab can potentially do something for you. As Dr. Farmer mentioned before she left, actually she said, you should tell everybody that bioengineering can solve all the problems. But I think maybe one day I can really confidently say that. At least now we can do something to make the change, make it better. Thank you for all the work that I present today is not done by myself. It's by a whole group of people. And we have a lot of surgical residents. We have postdocs. We have graduate students. We have project scientists. We have technicians, undergrads, and many different students and technician people. Very appreciate all those help. We have all these fund sources. This is not the most updated picture of the surgical bioengineering lab. We have the lab in the research 2 building. So if you have time, just stop by and I want to talk to you more about anything. Okay, this is my email. Thank you very much.
Well, a tour de force summary of bioengineering and scaffolding and stem cells. Thank you very much. How many surgery residents or fellows are in the lab at any one time?
So at this point, we have 3.
3.
Yeah, usually 2 to 3.
Yeah.
Yeah, different years.
And any medical students, undergrad students?
Yeah, so I have trained many, we have many pre-med students who are interested in applying for med school. Yeah. They want to get some research experience. We also have trained some medical school students, summer research projects, those kind of things.
Any questions?
Dr. Brown?
With the stem cell approach, the challenge is always getting approval because of the risk of cancer.
Right.
As you say, and you've shown with your histology and your in vitro stuff about the potential success of paracrine or the secretome aspect of it. Is there any concern that the secretome changes in the context of being in vivo? And if so, are you able to perhaps recover your matrix over the first 2 weeks of placement, and then use like chromatograph to essentially get a separation of what that secretome looks like over that 2-week period?
Right.
That would give you potentially the complete cell-free package that
right.
works.
That's a fantastic idea. So, as you mentioned, very true that stem cell product is very great, but you have to go through FDA approval, very tedious, and you need to have a GMP facility to get this live cell product, and there are many safety issues with that. So what we're trying to do, we're doing stem cell product very actively now, but on the other side, we really want to develop some cell product, a cell-free product, so that can hopefully mimic the cell product. As you mentioned, very true that after you transplant live cells in vivo, at different time point, the cells are changing. They are changing. I think they're really changing, especially, they die off over time. They must be changing significantly. So we cannot really control the cell fate once you inject or transplant the cells in vivo. But I think if we really want to develop a accurate mimicking for the live cells, you would have to go in there and look at different time points probably. Exactly. That would be great. But I think that's doable because the injectable or infusionable exosomes, for example, they're just like a drug. You can inject at different time points, right? If you know how the live cells are behaving in vivo at different time points, you direct that product appropriately and you inject them during that. That would be a very fantastic approach.
Scott?
Yeah.
Are you worried about retroviral contamination of the exosomes? And if so, what are you doing to mitigate against that?
Yeah, well, that's a great question. So as far as I know, in the U.S. right now, actually, there's no FDA-approved product yet. But in the Europe, actually, they have some clinical trial going on already about this. But so far, I haven't seen very significant adverse side effect of the exosomes for the viral contamination or anything. But I think that's certainly a very important thing to look at.
Some sort of primitive screen for retroviral
right.
segments would be pretty
yeah.
useful.
I haven't seen any publication actually. I think this is a very new field. Well, exosomes have been described for many years, but for regenerative medicine application, just a few years old. But now actually many people are in this field trying to develop this as a product. But I think many things need to be learned actually before we can really translate. Dr. Boy. Thank you.
Bioengineering role is no question and there's so much in your presentation. I'm just going to point in just one very small aspect. That is, I'd like to know your thoughts on the role of age ECM on how it affects the interactions.
Yeah.
Just as an aside, I think senescent cell telomeres, no longer divided.
Right.
If you think senescent cells on young patients, guess what? Telomeres regrow, reverse.
Right.
This is huge.
Yes.
So
Yes.
a lot of research has been done that our biology concluded the age of the matrix we use impacts the interaction with the adult cell and older cells that come on.
Right.
That's just a comment. I'm just saying it's going to be important. You're looking at the relationship, but I'm just saying you're in a very unique position because you have access to very young matrix.
Right.
Great.
The youngest matrix.
Hydrogel matrix. We do know, we talked about this
Right.